everyone, welcome back to your channel Chemistry Simplified. Today we are back with one more important topic of Biomolecules class 12 chemistry, that is protein. In this video we will be learning about the proteins and which make them, that is amino acids. We'll also study about the structure, their classification and denaturation of protein. Okay, so let's get started with the heading protein. Protein. What is protein? First of all, the question. You know that proteins are the biggest or we say the most important part of our life, right? With the cheap sources of protein like milk, egg, dal, cheese, meat, fish, etc. We often see, right? Often hear whenever you go to doctor, they ask you to take more and more of proteins and all, okay? But chemically, what do we call protein? So here, the word protein has been derived from the word proteus, the Greek word proteus, which means primary or of prime importance. Basically, these proteins are the polymers of alpha amino acids. Now, what is amino acids? Amino acids are the building block of proteins, and it contains two very important functional groups: the basic amino group, that is NH2, and a carboxyl group. And depending upon the position of this amino group with respect to the carboxyl group, it can be alpha, beta, gamma and so on. But since we get on hydrolysis of a protein, we only get alpha amino. That is why we study alpha amino acids. Okay, and these alpha amino acids, they are named depending upon their properties. For example, you have glycine, which is derived from the word glycose, Greek word glycose, which means sweet in taste. Meanwhile, triosine has been obtained from cheese, and the word is derived from Greek word trios, cheese. So on their, depending upon their origin, on the properties, they are named as accordingly. And generally we represent an amino acid by a 3-letter. For example, like glycine can be represented by a 3-letter GLY, glycine, or alanine by ALA, or leucine by LEU, and so on. Okay, so the general formula for an amino acid can be written as a carboxyl group and NH2 group. This is H, and here it is R, which is the side chain. Okay, so this is the introduction of amino acids. Okay, next we will see the classification of amino acids. I'll shortly take notes, or you can take screenshot if you want. Okay, so alpha amino acids can be further be classified in different ways. First, based on the R group. Based on the R group. For example, it can be non-polar, that is hydrophobic. Example we have is alanine, which is having a R group. I'm writing it here. If we replace the R with a CH3, then it is a non-polar hydrophobic amino acid, and we can have a polar, that is hydrophilic example we have is threonine, or we can also give example of serine. If we replace this R here with okay, so it will be a hydrophilic part. Okay. Then it can be acidic or it can be basic. Acidic example, you can write aspartic acid. That is H, and basic arginine. Okay, so this is on the basis of R group. Okay, second classification is based on nutrition. Based on nutrition, it can be classified as essential and non-essential. Essential and non-essential. Essential amino acids are those amino acids which cannot be synthesized by the body, जिसे हमारी body synthesize नहीं कर सकती है, and we have to take it from the diet. हमें हमारे diet में उसे include करना पड़ता है. ऐसे amino acids को हम essential amino acids कहते हैं. For example, like what can be? From here you can see arginine. It's an important one. Okay, leucine. All these are essential amino acids. In your NCERT book, there are 20 essential amino acids, out of which you can see all the asterisk marks. They are the essential amino acids, and you can write arginine. Okay, non-essential amino acids like aspartic acid, glycine, alanine. Non-essential amino acids are those which are produced or which can be synthesized by the body, जो हमारी body already synthesize कर रही है और जिसे हमें दूसरे से लेने की ज़रूरत नहीं है, वैसे amino acids को हम non-essential amino acids कहते हैं. For example, we have already written one is alanine or glycine. Aspartic acid. Okay, there are many more other examples. We have histidine, tryptophan, methionine, then threonine, arginine, isoleucine. All these are the essential amino acids, and those अमीनो एसिड्स जिसे हमारी बॉडी प्रोड्यूस करती है एंड विच वी डू नॉट नीड टू टेक इट इन आवर डाइट, दैट आर नोन एज non-essential amino acids, whereas those amino acids which are not synthesized by the body and have to be taken by the into our diet, they are known as essential amino acids. So amino acids can be classified in two ways. Based on the R group, it can be hydrophobic, hydrophilic, acidic and basic. Okay, then we discuss the properties of amino acids. You can take screenshot. Okay, then we have next is the properties of amino acids. Amino acids are basically colorless, crystalline solids. They are soluble in water and have high melting. They are high melting solids. Okay, now they behave as salts. Why do they behave as salts? Due to the presence of these two functional groups, that is amino group and the carboxyl group. Now a very important question that is often asked, that is known as Zwitterionic. What is Zwitterionic? Or let us understand the amphoteric nature or behavior of amines. Amphoteric behavior of protein. Just a moment. Okay, so in order to understand, here ये आपका कार्बोक्सिल पार्ट है, ये हमारा अमीन पार्ट, ठीक है. What is the amphoteric behavior of amines? First of all, we know that acids are proton donors and bases are proton acceptors. So in aqueous solution, what happens? In aqueous solution, this acidic carboxyl group, it loses a proton. It loses a proton, whereas this amino group, it gains one electron. The carboxyl group, यहाँ पे क्या है, यहाँ पे हमें पता है कि carboxyl group जो acids होते हैं, they are proton donors. तो वो क्या कर रहा है, यहाँ पे carboxylic acid एक proton lose करता है और amino group which can accept a proton, it is acting, it will take a proton, which makes it behave as a dipolar ion. For example, you can write it like this. So here it will behave as a dipolar ion, also known as the Zwitterionic. A question which is often asked. Dipolar ion or a Zwitterionic. Why? And this is neutral because of the presence of both positive as well as negative charge. The negative charge and the positive charge. This is very important question. This chapter is again important, as I already told you. Amino acids, they are colorless, crystalline solids, they are soluble in water and they have high melting point. They behave as salts due to the presence of the carboxyl group and the amino group. Now, the most important thing is that the amphoteric, these amino group, they have amphoteric behavior. Why they have amphoteric behavior? Because here we know that the carboxyl group, okay, they have the tendency it can lose a proton. So it will lose a proton. Meanwhile, the amino group can accept a proton, which makes it a dipolar ion. Okay, this dipolar ion, or also known as a Zwitterionic. Okay, so this is the property of amino acid. Now, I hope you are understanding this much. Let's move on to the structure of protein. Okay, structure of protein. Heading is structure of protein. We know that proteins are the polymer of amino acid, alpha amino acids, and they are connected by these proteins are the polymers of amino acids, and these amino acids are connected to each other by a peptide bond or a peptide linkage. Now this peptide linkage, what is that? This peptide linkage is nothing but a amide formed between the carboxyl group of one molecule and amino group of another molecule. Okay, let us understand by an example. Here we have an example. And another one is, this is one. Another example we have is we have these two, glycine and adenine. Okay, so in this case, what I told you, there is a formation of a peptide bond between the carboxyl group of one and amino group of the other molecule. Here, when these two will combine with each other, there will be a loss. This we can write it like this. I'll write it like this. Okay. This combination will remove a water molecule from this position. Removal of a water molecule will give rise to an amide bond. So this is okay, and this is known as glycyl adenine, that is glycine. Okay, so this is how the structure of protein is made. Proteins are the polymer of amino acid, and the amino acids, they keep on combining with each other. One amino acid combines with another amino acid, combines with third another amino acid, and amino acid, and amino acid, and so on by a peptide bond. And this peptide bond takes place by the reaction between the carboxyl group of one molecule and the amino group of the another molecule, and there is a removal of a water molecule giving rise to a polypeptide. Here two amino groups have combined together, so it is a dipeptide. We can call it as a dipeptide. If a third amino acid joins together, then it will be tripeptide. Similarly, tetrapeptide, pentapeptide, and so on will lead to a polypeptide. Now, if there are so many polypeptides, if one such polypeptide will have around more than 100 polypeptides joined together with a molecular mass of 10,000 units, then it is known as a protein. Then it is known as a protein. Okay, I hope this is clear to you. Now, this protein can further be classified. Proteins can further be classified as fibrous protein and globular protein. What is the difference between these two? Fibrous proteins, when these polypeptide chain, when they are held together in parallelly by the hydrogen, when these polypeptide chains are held together parallelly by hydrogen and disulfide bond in such a way that they give rise to fiber-like structure. Fiber-like structure, then it is a fibrous protein. And this fiber-like structure, it is insoluble in water. One such common example is our hair, that is the protein of hair, that is keratin, which is present in our hair, nails, wool. Okay, so you might have seen there are a lot of hairs everywhere. Even if you go to washroom, you see a lot of hair. Hair in the washroom, but they never dissolve. Okay, because they are insoluble in water. Then another example is myosin. Okay, then what is a globular protein? Now, when these peptide chains, when they just coil around each other, then they form spherical structures which are soluble in water, are called globular protein. For example, we have insulin, or we can say albumin. These are the examples of globular protein. So what we have seen here is proteins can be classified on the basis of shape as fibrous protein and globular protein. Fibrous protein, they are where the polypeptide chains, they are held parallelly to each other by hydrogen and disulfide bond, and they give rise to fiber-like structures which are insoluble in water. Example, keratin, myosin. Then globular protein, where the polypeptide chains just coil around forming spherical structures and they are soluble in water. Okay, example, insulin and albumin. So I hope this is clear. Please take a screenshot and then we will move on to the last part of the video. That is the structure and shape of protein can further be studied in 4 different levels. That is, first one is 4 different levels. First is the structure of protein can further be studied in like this: the primary structure, the secondary structure, the tertiary structure, and quaternary structure. Quaternary structure. First one is primary structure of protein. Now we know that these proteins, they can have one or more than one amino acids. Okay, and these amino acids, they are bonded to each other. Okay, and they are bonded to each other in a specific sequence. For example, suppose this is a sequence A. We know that, हम जानते हैं कि प्राइमरी स्ट्रक्चर प्रोटीन क्या होते हैं. प्रोटीन्स आर द पॉलीमर्स ऑफ अमीनो एसिड्स, एंड दीज अमीनो एसिड्स दे आर कंबाइंड टुगेदर with a proper link to each other, in a proper sequence. Okay, now this proper sequencing, this specific sequencing of the amino acid is the primary structure of protein. So if there is any change in the primary structure, then the sequence will change, and the sequence will change, it will give rise to a new or different protein. So what is primary structure? Primary structure gives the specific sequence of the amino acid. Okay, then comes the secondary structure. What is secondary structure? The secondary structure basically tells us about the secondary structure tells about the shape. What is secondary? It tells us about the shape in which the polypeptide chain can exist. Basically, they can exist in two ways. Okay, one is the alpha helix form. Second is the beta pleated structure, sheet structure. What is the difference? The difference is, in alpha helix, the structure is like this. Here, suppose there is a structure. I'm just trying to show. In alpha helix, there is a coil-like structure like this, and there is a bond between these two. They are bonded by a hydrogen. This occurs due to the regular coiling of the backbone of the polypeptide by hydrogen bonds of the carboxyl group and the amino, between the carboxyl and the amino group. So there is a bond with a hydrogen bond between these two. Okay, that is alpha helix. So this alpha helix structure, how it is occurring? It occurs due to the regular coiling of the backbone of this polypeptide by the hydrogen bond. And in beta, they run parallelly like this. So it is A. Beta plated, they run parallelly like this, and there is a hydrogen bond between them. So it is HH like this. Okay. Like this. So this is beta pleated and this is alpha helix. The third is tertiary. What is tertiary? The further coiling of this secondary structure leads to the tertiary structure, and the stability of this 2 degree and 3 degree is maintained by 4 forces. Basically by the hydrogen bond, then we have disulfide linkage, van der Waals forces, and electrostatic force of attraction. and the last one is quaternary. So some of the proteins, they exist in one or more polypeptide units, okay, and the spatial arrangement of such units, that is the quaternary structure of protein. So we can simply writing it the spatial arrangement of polypeptides give rise to quaternary structure. Okay, so this was all about the different structure and shape of protein. The last thing is denaturation of protein. What is denaturation of protein? You might have seen at home when you boil egg. What happens to the egg? जब आप egg लाते हैं, egg is very fragile, right? So when you bring egg, it is very fragile, but when you boil it, it becomes hard. What is this phenomenon? You take milk, and overnight you can see. The next morning you have a curd. How does it occur? What is the phenomenon? So basically, the protein that exists in the biological system has a very unique 3D structure, and that is said to be in its native form. Okay, but when this native form is subjected to any change like physical change, okay, if there is any change in the physical state like change in temperature or any change in the chemical change such as change in the pH, then what happens? The biological or activity of this protein gets disturbed. Okay, basically what happens to the alpha helix? They get uncoiled. Okay, they get uncoiled, and this what happens, the helix get uncoiled, the globules, they just get disturbed, and due to this what happens, the structure gets deformed. Okay, so the secondary structure, the tertiary structure, they gets deformed, and because of this is what is called denaturation of protein. And the examples of denaturation of protein is the egg albumin, that you see when the egg is boiled, it gets hardened. Okay, so on this I have a question for you, that is, where does the water present in the egg go after boiling the egg? Okay, so you find the answer and you can put your answer in the comment box. Okay, so I think we can give a recap. So what we have learned in this today's class, we learned about the proteins, amino acids, what are essential amino acids, what are non-essential amino acids. Essential amino acids which are required in the body through different diet because they cannot be synthesized in the body, whereas non-essential amino acids which are synthesized in the body and cannot be, we don't need to take it from the others. Then we learned about the structure of protein, how these proteins are the polymers of amino acids, and they are bonded by a peptide bond. Then there are two types of proteins: fibrous protein, globular protein. And lastly, the primary, secondary and tertiary. Primary structure of protein gives the specific sequence of the protein. Secondary structure gives the shape of the protein, that is basically alpha helix and the beta pleated structure. Then the stability is maintained by the hydrogen bond, the disulfide linkage, the van der Waals forces, as well as the electrostatic force of attraction. And then we have the quaternary structure of polymer, which is basically the spatial arrangement of this polypeptide units. Then last is denaturation of protein. Whenever this native protein is subjected to any physical change or chemical change like change in pH, change in temperature, then they lose their biological activity, they get deformed. Okay, but the structure of the primary structure of protein is not affected by it. Okay, so I think this much is clear to you. If you have any doubts, do let me know. I hope you like my video. If you like it, do not forget to like, subscribe and put on the bell icon. Thank you so much. See you in the next video.